So our project is looking at injectable hydrogel scaffolds for bone fractures caused through osteoporosis. My name's Luke.
I'm Colin.
I'm Matt. And I'm Mike.
So the first thing we did was identify a clinical need for our product. Every year, osteoporosis affects 1 in 3 women and 1 in 5 men over the age of 50, and it leads to approximately 8.9 million bone fractures. On average, bone fractures take approximately 6 to 8 weeks to heal through their bone tissue and the bone growing back. Problems with current bone fracture surgery include irritation, discomfort at the procedural site, the cost of the surgery, and the invasiveness. So currently, autografts are seen as the golden standard for bone surgery, but there are many limitations to this procedure, such as how invasive it is and the lack of donors that will supply the autograft. Some current hydrogels used on market look at natural polymers and synthetic materials, but there are problems with natural polymers in that they lack poor mechanical strength and they can only be applied to non-weight-bearing sites. Synthetic materials have often shown problems with biological patterns, and they often produce acid byproducts. So we really found a need, that there is a need for injectable hydrogels that mix these two products together.
So in order to overcome this problem, we decided to go with a silica-based bioactive glass reinforced gelatin hybrid polymer, which is depicted in Figure 2. This has advantageous mechanical properties such as strength, compressive strength, has better control to degradation and mineralization. It also mimics native bone tissue and has high osteogenic activity. Also, we would like to encapsulate bone marrow mesenchymal cells into our hydrogels to further increase the bone formation. And then finally, we would like to functionalize our hydrogel with alpha-2 beta-1 specific peptide, which when added into critical defect sites has been shown to protect encapsulated BMP2, attract osteoprogenitor migration, as well as promote mechanical bone formation. So we believe that by incorporating these 3 aspects into one scaffold, we'll be able to create a less invasive but more effective hydrogel that can be injected directly into the defect.
Although we have not pinpointed an exact price for this technology, we realize research and development stage will be quite costly because we're mixing multiple technologies into one product. Also, it will be costly to encapsulate bone marrow mesenchymal cells and other bone growth factors into one scaffold. The benefits of this product is that it's less invasive, promotes bone growth. It also eliminates corrosion and stress shielding because it eliminates metal from the surgery, and also has shorter hospital stay times for the patient.
Some alternatives that are currently out on the market are autografts, as mentioned earlier, as well as allografts, and bone matrix or bone graft substitutes. Autografts and allografts are very invasive as they require 2 open surgical sites, which requires a full surgical team as well as a long stay at the hospital, and therefore can be very expensive. This is shown through a study which showed patients were charged with almost $5,500 for an allograft and almost $5,000 for an autograft when involved in reconstructive surgery. Patients also experience extreme pain and discomfort following the surgery, as well as up to several years later can still experience pain from the site location. Bone matrix and bone graft substitutes are also out there and are slightly less invasive as there's no second site surgical procedure. There's only the first site. However, this still requires a full surgical team as well as a stay at the hospital, and can also have debris from material degradation. One study was concluded that a localized injection would be a viable option, an alternative, which is something that our product would be able to handle.